I'm George Pollack. I'm professor of neurobiology here at the University of Texas at Austin. In this movie, I would like to tell you a little bit about the research that I, together with my graduate students and postdoctoral fellows, have been conducting on the brain over the past 20 years at the university. The studies in my laboratory are concerned with how the brain processes sound and how acoustic features of the external world are represented in the brain. Or in other words, what happens to sound when it stops being sound and turns into neuronal activity? The subjects of my experiments, however, are not humans, but rather are bats. There are two reasons that I use bats to study the auditory system. The first is that I know the acoustic signals that bats use in their daily lives, which I then use to study their brains. The second is that the auditory portions of their brains are greatly enlarged, and they're enlarged because they rely on hearing to a greater degree than any other animal for survival. The enlargement, in turn, provides numerous experimental advantages which we exploit in our studies. Of course, bats are best known for their ability to use a form of biological sonar called echolocation. They use echolocation not only to orient in the environment, but even more importantly, to hunt insects and capture their prey. Here, I'm going to show it to you in this next movie. Check it out because it's really cool. In the evening, the bats leave their cave to hunt insects in the night sky, as we just saw. But during the day, they live in the cave, where they congregate in large colonies that often number in the millions. Let's go in the cave and let's check it out. It's pitch black and you can't see anything. And that's the point, because neither can they. In the darkness, vision is of no value. But we have lights, so let's turn them on and see what's going on. There are millions of them, but they just don't hang around all day. Rather, they have a social structure. Mothers have to communicate with their pups. Males have to establish territories, and they have to attract mates. These and a host of other social interactions are established and maintained largely through vocal communications. But it's in the lab that we study the various communication signals that the bats emit. Here, we made pouches. During the breeding season, each dominant male claims a pouch as his territory. We know each bat, and we know the pouch that each bat claims. The courtship behavior of males is one of the prominent examples of the social interactions that the bats engage in. This is Sid the bat. Sid is on the make and attracts females into his harem by wooing the ladies with his courtship song. The song is elaborate and complex. Let's listen to it so you can hear it for yourself. The question we ask in our experiments is, how does the auditory system enable Sid and all other bats to recognize one call and distinguish that call from all other calls? We are particularly interested in how excitation and inhibition interact in the auditory system to generate neuronal selectivities and thereby allow neurons to respond only to some signals and not to others. We use three principal techniques to make these evaluations. With the first technique, we present various notes or elements of the communication calls and evaluate how neurons respond to those calls before and while inhibition is blocked by the iontophoretic application of drugs such as bicuculin and strychnine. In the example shown here, a neuron in the auditory midbrain fired action potentials in response to only 2 of the notes and failed to respond to 8 of the notes. When inhibition was blocked, however, the same cell now responded to 8 notes, showing that inhibition acted to selectively shape the neuron's responsiveness to natural signals. The second technique is a computational method whereby we present a wide variety of signals and use the responses evoked by those signals to generate spectrotemporal receptive fields. The spectrotemporal receptive fields are also called STIRFs for short. The STIRFs are supposed to represent the frequencies that evoke excitation and those that evoke inhibition, as indicated by the arrows. But they also infer the relative timings and their magnitudes of excitation and inhibition. A two-dimensional representation of the STIRF is shown in the top left, and a three-dimensional representation of the same STIRF is shown in the lower right. The way the two representations relate to each other is seen when the two-dimensional STIRF is first rotated so that frequency is now on the abscissa, and then the STIRF is moved and superimposed on the three-dimensional STIRF. Now the colors that indicate magnitudes of excitation and inhibition form the third dimension. If the spectral temporal receptive field actually represents the true features of the neuron, then it should also predict how the neuron should respond to any signal, including communication calls. For example, this is a spectrogram that shows 4 notes in the chirp syllable of the courtship song. Frequency is represented on the y-axis, and time is represented on the x-axis. The colors show the amount of energy in each of the frequencies, where blue indicates high energy and red indicates low energy. The first 3 notes are simple downward sweeping frequency modulations, or FM sweeps. The 4th note is a little bit more complicated, consisting of a downward FM, then an upward and a slightly downward terminal component. This is what the notes sounds like. The spectrotemporal receptive field, or STIRF, can predict the responses to any complex signal through a process called convolution. Convolution, in essence, determines how closely the acoustic features of a signal match the acoustic features of the STIRF at each point in time. The closer the match, the larger the predicted response. And here's how it works. First, we get rid of the extraneous noise in the spectrotemporal receptive field, and then we flip it. Now we obtain the predicted response by marching the STRF across the spectrograms and multiplying the magnitude of the STRF with the energy in each portion of the spectrogram. There's the predicted response. Finally, we can compare the responses predicted by the STRF with the responses that were actually evoked by the 4 notes. In this case, the correlation between the predicted and the evoked responses was 0.8, and thus the predictions were very, very accurate. But the STIRFs can be convolved with any signal, including a whole host of other calls as shown here. And the predicted responses to those calls are as good as the predictions for the 4 notes of the chirps in the courtship song that I just showed. The third technique is called in vivo whole cell recordings, which I will explain to you in just a moment. Here we evaluate the most basic element or note in each of the communication and echolocation calls. Each call contains frequencies that change over time and are called frequency modulated, or FM sweeps. FM sweeps either sweep downward from high to low frequencies and sound like this, or other FM sweeps sweep upward from low to high frequencies and sound like this. With in vivo whole cell recordings, an electrode is inserted into the brain and positioned over an individual neuron.
The electrode makes contact with the membrane of the neuron and then sucks the membrane into the lumen of the electrode and breaks the membrane. The electrode thus has access to the interior of the neuron and records both the inputs to the neuron, as reflected in the changes in the membrane potential, the excitatory or inhibitory postsynaptic potentials, and the output of the neuron, the action potentials or spikes that are evoked. We can then present a downward FM sweep and record the responses evoked by that signal. The excitatory postsynaptic potential, or EPSP, is shown, as are the spikes. But we can also calculate the underlying excitatory and inhibitory conductances that generated the response. The conductances show the magnitude and the timing of the excitation and inhibition that played upon the neuron due to the signal that was presented. We can also present an upward FM sweep. For comparison, I show the responses and conductances evoked by the downward sweep, but now the upward FM is presented. Notice that it only evokes a small subthreshold EPSP, and thus there are no spikes evoked by the upward FM. Below, the conductances evoked by the upward FM are shown. Notice that there are prominent differences in the conductances. The excitatory conductance evoked by the downward FM, which produced a larger response, is larger than the excitatory conductance evoked by the upward FM. The reverse is true of the inhibitory conductances. The inhibitory conductance evoked by the downward FM is smaller than the inhibitory conductance evoked by the upward FM. Finally, notice the difference in the timing of the conductances. Excitation was evoked first by the downward FM, which evoked a larger response, whereas inhibition was evoked first by the upward FM, which evoked the subthreshold smaller response. So there are differences in both the timing and magnitudes of the excitatory and the inhibitory inputs or conductances. But what are the relative contributions of timing and magnitude? We can actually determine the contributions by manipulating the conductances and then computing a response from those manipulated conductances in a model. The modeled responses are then compared to the responses actually evoked by the FMs, and thus the degree to which each manipulation changes the response can be evaluated. For example, we could ask what would be the effect of just reducing the magnitude of the excitation for the response to the downward FM. Alternatively, we can ask what response would be evoked if in the downward FM inhibition led excitation rather than following it. To what degree would such a timing difference influence the response? Would the change in the response due to changing the timing be as great, or would it be less than a change in magnitude? In this way, my lab is evaluating not only how the inputs that play upon auditory neurons shape their selective response properties, but which of the input features is the principal determinant of the cell's response. The ability to obtain responses and the parameters of the underlying conductances that generate the responses is one of the most powerful methods for determining how the nervous system creates new or emergent response properties in higher order regions of the brain. So thanks very much for joining me, and I hope you learned something with this brief tour of the research in my laboratory.